Here's another way of measuring it. Instead of in colors, we have z-scores, and underneath there's regions of interest. There's about 10 region pairs in their left hemisphere because this was the language task. And you'll notice the top line is for controls. That's their functional connectivity between those region pairs. Below that and parallel is the curve for the autism group. Now, the importance of that is that it's not a totally different pattern. It is a similar pattern of connectivity, but just less than. And with developmental studies, we've shown that it does make progress with development, but just not nearly enough. And that's in those we can test, which would be the high-functioning individuals. Here's another study that you'll see has left hemisphere and right hemisphere pairs. It was an executive function test, Tower of Hanoi, and you see the same pattern of under-cortical connectivity. The next thing that I mentioned, though, is outcome, the genes. We have a rather long list. This is from Abrams and Geschwin's paper in 2008, which is a superb summary. And here we have syndromes. Let me see. Yeah. These are some of the genes that have been reported. You'll probably see there's fMRI1, SHANK3, the neuroligands, neurolexins, things that you've heard about for years now. And then, of course, the ASD-related syndromes. So from a geneticist perspective, we then had non-syndromic autism, which is those cases that didn't have a phenotype or features that they recognized as a syndrome, and we have syndromic autism. So if you hear people saying, oh, syndromic autism, they're talking about the list over on the right side. But the interesting thing is if you look at the genes in the areas next to, say, the 15q or the 22q, you see UBE, you see SHANK3, fMRI1, MECP, and those genes all appeared on the first list. So we begin to understand why individuals with autism who have no obvious phenotype at this point that distinguishes them have some of the same genes that you see under syndromic autism. So at least those are starting to come together to make sense. Now we have a long ways to go before we know what each of those genes are doing and what's not happening in autism. I think the next thing was the reports of copy number variation, and here you have the chromosomes, top left, chromosome 1, all the way over to 11, and then it continues on the bottom, 12 to 22 with X and Y on the end. And the little pinkish red areas are the areas where they've seen what they call copy number variation or failure to accurately reproduce the DNA from the previous generation to the current generation, so to speak, okay? The thing that was remarkable to me about this is they're scattered. You know, we might like to think that everything's nicely organized like a library and they put all the autism genes, so to speak, in one chromosome or maybe two. But they didn't, that's not the way it works. Scattered throughout. And that, I believe, explains to us why we can have different chromosome syndromes from 13 to 15 to 16 to 21, all of which can manifest signs and symptoms of autism. The genes that contribute to autism are scattered. And the other thing you want to keep in mind is that all of the DNA, which is the architectural plan for every organ in your body, is contained in the egg and the sperm, and then they fuse. All of it is there. Each of you started as that one cell. And the amazing complexity there is in that DNA instruction, set of instructions, with only 30,000 genes. So that means each gene has to do a lot of things, and it does different things at different points in development. And when you get to look at the complexity, you're not amazed that there are errors in brain development, which I would call a neurologic disorder. You're amazed that you have so many people that don't appear to have impairments. How can the brain develop, and the body normally, so very often? Now, a couple of years ago, and I may be off by 1 year on this, the geneticists and the clinical researchers got together and talked about, well, where was autism now? And the theme of the conference was, is autism a synapsopathy? And a synapse is the connection, the contact between neurons. We'll keep it simple. And there can be millions and millions of these connections. So the issue was, at that time, there were enough advances, they thought, well, is it about synapses? But the other thing that I saw at the meeting is that the geneticists were kind of over here and had a lot of knowledge about the genes and some about what they might do in an animal model, but they didn't really know very much about the clinical syndrome, so they weren't really able to relate one to the other. And then there were the clinicians on the other side who understood a great deal about autism and were having trouble relating that, what they knew about autism, to the synapse. And so the issue is, how do we put this together to the extent that we can? Science is steadily evolving, and we know so much more than we knew 5 and 10 years ago. But we only know a small fraction of what we need to know about the pathophysiology and the etiology of autism. What I would say is that anyone who talks about the cause of autism needs to be able to think about how all these pieces fit together, or you will not give very accurate advice about the cause of autism. The other part of that is that if you can't think about all these pieces, then maybe you should not talk about the cause of autism. We have dentists in our country telling parents what causes autism, which is beyond me, why they would even think they could do that. But I think it's very important that everybody knows they have a responsibility to know about progress in all of these areas in order to talk about cause. And something that Sally Rogers said in her paper in 2009, we need fewer nice stories, fewer fairy tales that sound good, but have usually turned out to have absolutely nothing to do with the biology of autism. And then, of course, I would advocate that we rely on science to tell us what we know and what we don't know. We're not at an answer. We're in the process. Science is the process of setting up an experiment with a design and with objectivity so that if somebody else does the experiment the same way, they will get the same result. Now, the next thing, of course, we have to communicate to the public and to families about what we're learning. They want to know, and in the absence of knowing, it can be dangerous. If you have a vacuum, something is going to fill it. People are going to try to figure out a reason for autism, but without all of the knowledge they need, they're not going to come to the right conclusion. And if they act on incorrect conclusions, they may well do more harm than any good. This is so complex, we're not just going to guess an answer. And then the other thing is, again, we need to integrate knowledge, because everything we learn at one of those levels will tell us something about adjacent levels. And that maximally improves our chance of making better treatments. Now, Didn't notice that one. So we're back to this sequence, is what we're about discovering. Now, how do we figure our way forward with those pieces of information that came out? It said the old way wasn't a good explanation, but what's the new way? And I think the best evidence for understanding the general nature of the cause of autism has come from the infant sibling studies. And Sally Rogers wrote an excellent summary paper last year in June in 2009. And she talked about what are the earliest signs of autism. At 6 months, the infants were normal. And then when the first manifestations emerged, they were subtle. They weren't marked, but they were in the first year. By 12 months, these studies were able to detect differences in sensory responsivity, under-overreactivity to sensory stimuli, unusual motor quality to the motor movements, and unusual visual regard, meaning what was it that interested the children. As our first speaker said, the children with autism would tell you how many lights there are in this room, but not necessarily know much about what I'm talking about. So that's what a lot of the children first presented with, was looking at ceiling fans or lights or things like that, things that other children were not. Now, by 12 months, there was delays on standardized testing. But the next thing that happened was between 12 and 24 months, all the other symptoms emerged. And that means not just the social or the gaze abnormalities, but the irritability, the temperament issues, the dysregulation of emotion, unable to calm themselves, the overactivity. All of these things emerged together as though they were part of the same process, which they are. And the other thing is some of the children had such a deceleration in development between 12 and 24 months that they lost half of their cognitive abilities. And what Sally concluded is that these findings do not support the view that autism is primarily a social communicative disorder, and instead suggest that autism disrupts multiple aspects of development rather simultaneously. Now, cause here means pathophysiology. It doesn't mean why is this child not interested in people, which may be that people don't have the salience that typical children are born with. In this instance, though, it's the brain and what's wrong with the brain. And many of the people that proposed a social impairment thought that the social deficit would still appear first in time, that would be absolutely hands down the first sign you saw, everything else would be the consequence of that. And that is absolutely not what they saw. It doesn't mean that you won't see reports of more and more subtle impairments noticed in these children, but this was the initial picture. So not a single primary deficit, but all of these things, including the associated signs and symptoms and the comorbid symptoms, were all emerging together as a result of the same neurobiologic process.